Today we have Lisa Ridnour talking. She got her Ph.D. at the University of Arizona. Iowa.
That's right.
And then you went to Arizona.
No, then I went to WashU.
WashU.
Yeah.
Okay. And then she came to NCI. She now is in the cancer inflammation program, so she works up in Frederick. She dared 270, and she got here early. So we're pleased to have Lisa. NOS2-derived NO mechanisms and disease progression.
All right, so today I'm going to talk about two different diseases, actually. I'm going to talk mostly about cancer, but I'm going to talk also a little bit about Alzheimer's disease, where we know that NO has an effect in disease progression. So to start out, talking about breast cancer. Breast cancer remains the second leading cause of death among women with malignancies, and diagnosis is generally determined by these classical indicators. Let's see. By these classical indicators on the top of the slide. But we think that we can improve disease management and survival by identifying new biomarkers that can aid in managing the disease and improving patient survival. And so, since we're going to talk about NO, the past 10, 15 years or so, the Wink Lab has spent a lot of time examining signal effects of nitric oxide, and they've used chemical donors that release nitric oxide to do this. And what we have found during this time is that at very low levels of NO, we get cyclic GMP-mediated effects. And as the concentrations begin to rise, we see HIF-1 alpha stabilization and phosphorylation of ERK kinase as well as AKT. Then at higher levels, we see p53 phosphorylation, and at even higher levels, around 1 micromolar steady state NO, that is consistent with nitrosative stress levels. So these intermediate and higher levels can be produced by the inducible NOS isoform NOS2. So we know that the components of the tumor microenvironment, we know that the tumor microenvironment is very complex, and it consists of cancer cells, immune cells, endothelial cells, and fibroblasts, as well as extracellular matrix. And all of these components within the tumor microenvironment communicate with each other to promote tumor progression. So Redox Biology of NOS-derived NO is a key mediating factor in these communication processes. And while over the years we've determined quite a lot about NO signaling in cancer cells, as well as the immune system and vasculature, less is known about NO signaling in extracellular matrix. And so when I first joined Dave's group, that was one of the first questions that we were interested in addressing. So we became involved with Dave Roberts here at NCI, and we began a collaboration looking at crosstalk signaling effects between nitric oxide and thrombospondin 1, which is a very large extracellular matrix protein, and it's generally thought to be anti-angiogenic, where in contrast we know that NO is pro-angiogenic. And so what this project determined, we used functional assays, including HUVEC proliferation and this vascular outgrowth muscle explant assay to look at NO effects and the negative effects of thrombospondin. And what we found was that at low flux NO, we see increased HUVEC proliferation, which peaks at about 1 micromolar. And in the process of inducing HUVEC proliferation, we see NO-induced suppression of thrombospondin 1 at these very, very low levels of nitric oxide. Similarly, in the explant assay, we see that in the control tissue, you see a small amount of vascular cells migrating out and away from the perimeter of the tissue. But if you add a NOS inhibitor, L-name, you blunt the outgrowth of these vascular cells. In contrast, if we add a little bit of nitric oxide to these tissues, we see very abundant outgrowth of vascular cells. And this is very consistent in this induced wound response assay. This work went on to show that all these processes can be inhibited by the addition of exogenous thrombospondin 1. So in summary, this project went on, it became a very fruitful collaboration, and it went on to generate a number of very nice papers for Dave Roberts' group. So another group of extracellular matrix proteins that we were interested in examining regulation in were matrix metalloproteinases. These are secreted proteases. They can process components of extracellular matrix. And the reason that we were interested in them is because the active site contains zinc thiolates, which can be targeted by and reactive nitrogen species. So we first started examining regulation of MMPs and their endogenous inhibitor TIMPs in macrophage cells that we exposed low doses of the donor sperno. And we can see that at intermediate concentrations of sperno, we see an increase in MMP activity, and we also see suppression of TIMP1 secretion at these lower levels. So we took the conditioned media from these cells where we saw peak activation of MMPs, and we went on and we put this media on this tissue explant assay. And what we found was that the media from cells that were treated with sperno at 10 micromolar showed, again, very abundant outgrowth of vascular cells away from the perimeter of the tissue bed. Then when we went on to knock down MMP9, we saw that this process was dependent upon MMP9 activity. So this work demonstrated that regulation of MMP9 in macrophages was in part cyclic GMP dependent, and it involved TIMP1 secretion, reduction in secretion of TIMP1, and increased MMP9 activity. So we wanted to then go on and look at this regulation process in physiologically relevant models. And so we proceeded to examine these effects in cancer and Alzheimer's disease. So before we talk about what we found in cancer, I'll tell you a little bit about what we found in an Alzheimer's model. And this model was developed at Duke University by a collaborator of ours, Carol Colton. And the mouse, basically, she made this mouse that overexpresses APP protein, which is a transmembrane protein. And when it's clipped, it can make A beta. Okay, so these mice, she then crossed these mice with NOS2 knockout animals. Okay, so she had mice that had functional NOS2 as well as mice that lacked NOS2 in all cell types that make it. Okay, so what she found was that the mice that lacked functional NOS2 had a lot of insoluble plaque, whereas mice that contained functional NOS2 did not. And importantly, when she looked at the state of the neurons in these animals, mice that had NOS2 did not exhibit dead neurons, but the mice that lacked NOS2 did have quite a lot of dead neurons when they compared to a head-injured control. So one thing that we know from the literature is that A beta is a substrate for MMP9, and this was demonstrated by a group at Washington University, and they showed that active MMP9 could clip A beta at these specific sites in the sequence. So we began to examine whether or not NO regulation of MMP9 and TIMP1 may be attributed to this difference in phenotype of the mouse. So the first thing that we did was we looked at MMP9 and TIMP1 expression in these mice, and you can see that in the animals that lack functional NOS2, they have lower levels of MMP9 expression, and they also have higher levels of TIMP1 expression. We examined MMP2 and TIMP2 levels in these mice, and they did not appear to have any difference in their expression levels. So then we went on and looked at in situ zymography of these mice, and so we used a gelatinase zymography assay. And so this would measure MMP2 and MMP9 activity. And what you can see is the animals that have functional NOS2 have much higher fluorescence, which indicates higher MMP activity in these tissues. And this activity seems to be restricted to MMP9 because we don't see really, we see minimal levels of MMP2 in these animals. So unfortunately, this part of the assay, the zymogram down here, wasn't able to give us any differences in activity levels. So we went on and we developed an antibody-based MRM assay to examine MMP9 activity in these tissues. And so we made an antibody that recognized a fragment of MMP9 clipped insoluble A beta. Many enzymes can degrade soluble A beta, but to the best of our knowledge, MMP9 is the only enzyme that can clip the insoluble form, okay? And it can clip it at these points in the sequence, and we made an antibody against the 1:16 fragment here. And this antibody was extensively characterized using mass spec. And so we used, to do this, we immunoprecipitated material from MMP9 degraded insoluble A beta, as well as the whole brain homogenate of the mice that contained NOS2. And we also used A beta 1:16 that we purchased. And what you can see is that these MRMs are all the same, and that tells us that this fragment is abundant in these animals, and you can measure it. And so we did that in the brains of the mice, which is shown on the top panel here, panel A. And what this shows us is that mice that contain functional NOS2 have a little bit more than twofold increase in the A beta 16 fragment. And we use that as an index of MMP9 activity. And these data suggest that these animals do have higher MMP9 activity, whereas in the absence of NOS2, it's reduced. We went ahead and we looked at human Alzheimer's brain and human control brain, and we see the same trend in these tissues as well. So together, these data tell us that perhaps nitric oxide is protecting against disease progression of Alzheimer's, in part by increasing MMP9 activity and lowering TIMP1, which facilitates the degradation and clearance of amyloid plaque to non-toxic peptide fragments. So now we're going to talk a little bit about cancer. And so on this slide, anything that is highlighted in green are cancer types that have published the ability of NOS2 to predict poor prognosis. And among them are breast cancer, which you can see on the right side of the slide. And so we went on to examine NOS2 as a predictor of breast cancer survival. We did a cohort study with Stefan Ams here at NCI Frederick. And what they showed in this study was that NOS2 did in fact predict poor survival in patients that had ER-negative breast cancer, which is a more aggressive form of breast cancer. In contrast, it did not predict poor survival in the less aggressive form of disease, which is ER-positive breast cancer. Interestingly, among the patients that succumbed to disease in the high NOS2 expressing group, 92% of those patients expressed elevated tumor NOS2. So another important thing that I wanted to show from this work is that NOS2 expression did correlate positively with tumor grade, with grade 3. It also correlated positively with p53 mutation, as well as CD31, which is an index of vasculature. So in this study, they used laser capture microdissection of high NOS2 and low NOS2 tumor tissues, and they did a gene array analysis, and they found a signature of about 44 genes that were upregulated in tumors that had high NOS2. And we were particularly interested in a set of these proteins, including IL-8, which is a basal-like marker, and it's involved in stem cell proliferation, angiogenesis, and cell migration and metastasis. In addition, the p-cadherin is also a basal-like marker. S100A8 is a toll receptor agonist involved in inflammatory processes. And CD44 is a stem cell marker, and these proteins were all elevated. Interestingly, I think this protein, arginine synthetase, this is very interesting to me because this protein will basically catalyze the recycling of citrulline, which is also a byproduct of the NOS mediated oxidation of L-arginine to form NO. Citrulline is a byproduct of that reaction. And this protein, arginine synthetase, recycles citrulline back to arginine, which may provide a feed-forward mechanism for keeping arginine levels high enough in the tumor to generate some NO. So I thought that that was very, very interesting. Anyhow, one thing that you don't see are MMP9 and TIMPs on this table, but there are some other connections here because MMP9 has been shown to process IL-8 to increase its bioactivity, and also CD44 can serve as a docking port for MMP9, so it would bind to it, and it's been identified at the leading edge of migrating cells, and it's been shown in some studies to be associated with increased metastases. Okay, so other groups have also demonstrated that MMP9 predicts poor overall survival and relapse-free survival, but TIMP1 does as well. So this can be thought of as a bit of a conundrum because TIMP1 is generally thought of as an inhibitor for MMP9, but it does have other MMP-independent functions. So we asked the question of whether or not NO might elucidate pro-tumor effects between MMP9 and TIMP1. So to do this, we went back to Stefan's cohort, and we stained for TIMP1 expression in these tumor tissues, as well as CD63, which is a cell surface protein that TIMP1 can bind to and initiate pro-survival signaling that goes through AKT. So what you see is that these tumors, these are just 3 examples, but we see differential expression in these tumors. Some had high, some had intermediate, and some had low or no levels of TIMP1 and CD63. So we went on and examined the survival effects of TIMP1 in our model, and we show that TIMP1 does predict poor survival in our cohort. And when we stratified for high NOS2, this effect seemed to be restricted to tumors that also expressed high NOS2 because TIMP1 did not predict poor survival in tumors that expressed low NOS2. So we thought that this might be telling us that these survival effects of high NOS2 and high TIMP1 may be mechanistically interrelated. So what might a common signaling pathway be? And AKT signaling may be commonly shared as a mechanism. So again, we know from the literature that TIMP1 can bind to CD63 to initiate pro-survival signaling that goes through PI3 kinase, AKT, and bad phosphorylation to inhibit apoptosis. And we also know from previous studies from Stefan's lab that NOS2 correlates with phospho-AKT, and that is shown by this odds ratio of 4.49. So an odds ratio describes the strength of an association between two molecules and the odds of that event occurring in one group compared to another. So again, tumors that have high NOS2 and high PAKT exhibit an odds ratio of about 4.5. So we then stratified for high and low TIMP1, and in tumors that also express high TIMP1, this odds ratio goes from 4.5 to nearly 13. In contrast, tumors that express low TIMP1, the odds ratio goes down by nearly half. So we then looked at TIMP1 correlations with NOS2, and we found that TIMP1 only correlated with NOS2 in ER-negative patients. So we went forward with our cell culture experiments using ER-negative breast cancer cells. And we treated these cells with an INO donor detaino, and we found that at high concentrations of NO, we see increased AKT phosphorylation and bad phosphorylation. But when we knocked out TIMP1, these phosphorylation patterns were diminished. And we also show that on the bottom of the figure where we plotted the normalized phosphorylation of PAKT to total AKT and phosphobad to total bad in the cells that had high TIMP1 and cells that did not. So we went on to look at the expression, whether or not NO is inducing expression of TIMP1 in these cells. And we could not identify a significant increase at these donor concentrations in TIMP1 expression. We do see a significant increase in protein secretion at 100 micromolar of detaino, but this isn't consistent with the NO concentration that induced phosphorylation of the proteins. So then we began to think that we were kind of looking at this wrong, because this is actually measuring the levels of TIMP1 secreted into the media. But if it has to bind to the cell at a cell surface protein, maybe we're, not looking at the right place here. So we then looked in the literature, and we found a paper, an interesting paper, where they made a mutant form of TIMP1 that would not inhibit active MMP9. And they found that this mutant TIMP1 protein actually bound to a higher degree to CD63 than wild-type protein. And in their system, it did affect the phenotype of their system. So we asked the question of whether or not NO can modulate TIMP1 function and TIMP1 CD63 colocalization. And to do this, we did a confocal study of control and treated MB231 cells, and the TIMP1 is in green and CD63 is shown in red, and you can see that the cells that got there's a lot more green fluorescence, indicating that TIMP1 is stuck to the surface of these cells. And then there does appear to be some co-localization with CD63 and TIMP1, as shown by these yellowish, orangish regions on the bottom panel here. So what might be happening? We know that TIMP1 we know that NO can cause nitration events of proteins, and so we treated recombinant TIMP1 protein with these same concentrations of NO donor, and then we dried the protein down and flew it on the mass spec, and we see that there is indeed a 45 Dalton mass shift in the protein that we exposed to NO, and this is consistent with a nitration event. And we identified 2 nitration events on 2 tyrosine residues in this protein. So we went back and we immunoprecipitated MB231s with TIMP1, and then we blotted for 3 and T, and we see that there is an increase in nitrous tyrosine in the cells that were treated with NO, and there seems to also be an increase in pull down of CD63 in these lysates as well. And similarly, we went back and we looked at PI3K signaling in these cells, and we see that NO induces an increase in PI3K phosphorylation at 500 micromolar, which is reduced when we knocked out TIMP1. So together, these data tell us that TIMP1 predicts poor survival in patients with high NOS2 tumor expression, that elevated TIMP1 augments NOS2 PAKT association in the breast tumors, and that if we silence TIMP1, we can suppress NO-induced apoptotic signaling in breast cancer cells. So then an interesting paper came out about 6 months before we published our paper. And basically this work, they also showed, identified high levels of TIMP1 nitration, and this was in macrophages, I think, that they exposed to LPS, so that you see very high levels of TIMP1 nitration in this model. And then when they went on and did a reverse zymography, they showed that this nitrated TIMP was not able to bind to MMP9, and you see high levels of MMP9 activity in this zymogram. And so interestingly, they used a mouse model or a math model to predict what tyrosine residues would be nitrated in their system, and they predicted the same tyrosine residues with their math model that we showed by mass spectrometry. So going back to our patient cohort, we also looked at MMP9 as a predictor of poor survival, and it does predict poor survival. It predicts the hazard ratio goes up a little bit in tumors that have low TIMP1 expression, but MMP9 does not predict poor survival in tumors that have high TIMP1 expression. So we know that MMP9, in addition to processing IL-8 and increasing its bioactivity, it can also process VEGF and increase its bioavailability, and it's been associated with increases in angiogenesis. And then again down here, where I told you that papers have shown that it can bind to CD44 and promote metastases, we see in our model that patients that have both high MMP9 and high CD44 exhibit an increased hazard ratio of 2.49. So that may be, there may be a similar effect in our breast cancer cohort as well, and involvement of NOS2. So can NO elucidate pro-tumor effects of MMP9 and TIMP1? So we think what this data may be telling us is that tyrosine nitration may shift TIMP1 signaling away from its MMP9 inhibitory function to favor CD63 pro-survival signaling, while maintaining MMP9 activity to promote other pro-tumor effects like promoting angiogenesis, cell migration, invasion, and metastasis. So we went on and looked at whether or not we could target NOS2 in a mouse model. So we injected mice with MB231 cells that have a GFP tag, and that way we can look at fluorescence of these tumors in the mouse. And then we treated the mice with we gave them water that had aminoguanidine, which is a pretty specific iNOS inhibitor. And you can see that the growth rate of these tumors is significantly less than the control mice that did not get the inhibitor. And you can see fluorescence of their tumors as well over here. But the most striking observation from this study was that the control mice had more fluorescence than the mice that their brains had more fluorescence, and their lungs. We don't show that on this slide here, but we did look at lungs. Mice that got treated with aminoguanidine had far less fluorescence in their brain tissue, which indicated that there was less metastatic events in the brains of these mice. And Julie went on and she looked at some of our biomarker expression levels in the mice, and she looked at IL-8, CD44, S100A8, which we showed in our table of our human tumor tissues. And she also looked at COX-2, TLR4, which is the agonist of S100A8, and IL-6. And in all cases, the mice that got aminoguanidine had reduced expression of these biomarkers. So an important question arose in review of the JCI paper of the tumor cohort, and they wanted to know if NOS2 was just high or if it was actually making NO. And so Julie went on to look at how NOS2 might be regulated in tumors. And so she used components of the tumor microenvironment, including serum starvation, serum withdrawal. She used hypoxia, and she used cytokines, which to mimic chronic inflammation in the tumor microenvironment. And she found that when she just simply treated the cells with serum withdrawal when compared to control, she saw much higher levels of NOS2 protein expression. And she also quantified nitrite levels in these samples as well. And she showed, I think, about a twofold increase in nitrite levels when the cells were exposed to serum withdrawal. If she exposed them to gamma interferon, it was very, very high. And then we took these cells that were exposed to gamma interferon and serum withdrawal, and she looked at TIMP expression, and TIMP is induced dramatically by interferon gamma. And when she IP'd TIMP out and then blotted for 3NT, she saw a little bit of 3NT staining, which was maybe reduced a little bit in the presence of aminoguanidine. So together, these data tell us that NOS2 can perhaps drive disease progression. It may be an indicator of a chronic inflammation within the tumor. It can induce cell migration, pro-survival signaling. It can induce tumor growth and metastases. Julie also showed that it can induce chemo resistance as well. So a very interesting paper then later came out to support our findings, and this was done by a group at MD Anderson, and the senior author was Jenny Chang, and she looked at NOS2 expression in patients that had ER-negative disease, and she found that NOS2 also predicted poor survival in these patients, and this was verified in two other cohorts that she examined. So she wanted to target NOS2 in mice, and she gave them a different inhibitor, L-name, which is a pan-inhibitor of the NOS isoforms. Again, these are MB231 xenografts, and you can see that the mice that got the NOS inhibitor has reduced growth, and they also have reduced luminescence. These cells were tagged with luciferase. They have reduced luminescence in the lungs of these animals as well. So again, this study supports ours that targeting NOS can reduce tumor growth, but it can also reduce metastasis as well. And so she went on to show that HIF1 is elevated in the controls, but it's suppressed in the animals that got L-name. And she proposed that a signaling pathway involving iNOS going through TGF-beta and increased HIF1 would promote EMT and tumor metastases. And so this is just a slide this is new data that we have where we treated MB231 cells with arginine, aminoguanidine, and also we titrated back in Detaino in the cells that got the aminoguanidine. And this is interesting because we just looked at trajectory of dye-labeled cells, and when you compare to the controls, there's a lot more events, and you can see longer trajectories in the cells that got NO. So that's just a little bit of preliminary data for a new project that we're trying to start. But together, I think both of our study and theirs suggest that the key event here is targeting metastases by NO. Okay, so we wanted to know if we could target NOS in conventional therapy. So we used a radiation model. So we had mice that have tumor-bearing legs, and we gave them 10 gray of irradiation. And we treated these mice either with aminoguanidine to inhibit iNOS, or we gave them L-name. And in this case, this is an immunocompetent mice. And the animals that got L-name exhibited very, very dramatically reduced tumor growth when they got L-name and irradiation compared to the irradiation control. iNOS inhibitor aminoguanidine did significantly reduce tumor growth, but the tumor growth delay induced by radiation, but it wasn't as effective as L-name. So we repeated these experiments in nude mice using HT29 cells and the SSC cells, and basically there was no augmentation of the radiation-induced growth delay, and this suggests to us that T cells are required for targeting NOS in this system. So let's see. Okay, so we went on and we looked at cytokine expression profiles in these tumors, and the tumors that got, let's see, the tumors that got irradiation alone exhibited an overall immunosuppressive signaling profile, but the tumors that got 10 gray and L-name expressed higher levels of pro-inflammatory cytotoxic cytokines. So IL-10 was one cytokine that really jumped out at us because it was upregulated early in the mice that got 10 gray, and it was suppressed when we gave L-name. And so we wanted to know if we could target IL-10 and increase the radiation-induced growth delay, and we were able to do that, which is shown here. So we went on to look at the tumors of these mice, and we isolated leukocytes from these tumors, and we found that in the animals that got the 10 gray irradiation and L-name, they had a higher number of CD8 positive T cells and a higher number of activated CD8 positive T cells as well. So this data suggests that if we target NOS in a radiation therapeutic model, we can reduce immunosuppressive cytokines, increase pro-inflammatory cytokines, and increase the number and activation of CD8 positive cytolytic T cells to improve the overall tumor response to radiation. So we have also looked at CD40 treated mice in this same model. CD40 will initiate a cytokine storm of pro-inflammatory cytokines, and we show here that if we give CD40 the day before we irradiate the tumor, that we can increase the tumor growth delay when compared to the radiation alone. And Giorgio Trinchieri has demonstrated that antibiotics can limit the chemotherapeutic effects of oxaliplatin. And so we examined whether or not antibiotics have an effect in our radiation-induced tumor growth delay. And similarly, we see that when we gave mice antibiotics, it did, in fact, diminish the tumor response to radiation here. So these studies have opened the door to a new area where we can examine targeting NOS or, targeting polarization of the immune system to improve therapeutic response to radiation. Similarly, Jenny Chang also showed that L-name potentiated the chemotherapeutic effect of docetaxel in mice. And you can see that the survival of these animals is greatly extended here. These data show that iNOS promotes triple-negative disease progression by modulating. They postulate that it's modulating cancer stem cells in metastatic potential. And again, they identified two new biomarker proteins that were high in their ER-negative breast tumors with high NOS2, and knockdown of these genes reduced tumor growth and metastasis in mice. Mutation of these genes promoted disease progression in patients, and both of these proteins were shown to be regulated by NO. So we are also interested in looking at other novel compounds that maybe can target cancer stem cells and HIF. One of our fellows in our lab has made a series of aspirin conjugated drugs where she stuck Dino, which is an NO donor, onto aspirin, and Ipano, which releases HNO, which is a reduced form of nitric oxide. Importantly, these compounds reduce the ulcerogenicity of aspirin, but also if we give them to tumor-bearing animals, both the Dino aspirin and Ipano aspirin was even more effective at reducing tumor growth in these mice. Interestingly, these drugs can increase oxidative stress and DNA damage and apoptosis, but they also can target stem cell biomarkers, and they can reduce angiogenesis. So another colleague of ours from Brazil, they are making some ruthenium drugs that will release HNO as well. These drugs are activated to release HNO by thiols. And so here we show that when we induce HIF-1 stabilization by Sperno, if we treat the cells with Luis's ruthenium compounds, we can suppress NO-induced stabilization of HIF-1. We think that these compounds may be suitable for ER-negative patients that have high NOS2 because, again, going back to our table, these patients express high levels of glutamate cysteine ligase, which is the rate-limiting enzyme in glutathione synthesis. And also, if these drugs can target HIF and maybe reduce angiogenesis, we also know in our cohort that increased vascularization has been identified in tumors that have high NOS2. So basically, in short, this ruthenium compound that is thiol activated may be a suitable drug for treating patients with this signature. So to end the talk, I think these data have shown that we can use redox inflammation to follow cancer progression and treatment. And basically, these studies have kind of started from bedside to bench and back to bedside because Jenny Chang is, they're going to begin a clinical trial using NOS inhibitors in combination with chemotherapy, including docetaxel. And in part, we think that NO is targeting cancer stem cells, and it's promoting metastases of cancer cells, and TIMP-1 nitration and increasing MMP9 TIMP-1 ratio may be, an important mechanism in this process. So a lot of people helped with all of this work. Stefan's group here at NCI have the tumor cohort, and they have examined tons and tons of protein expressions in these tumors that we can go back and look mechanistically at relationship with NOS2. We have active collaborations with Dr. Wiltrout and Giorgio Trinchieri to examine roles of immune polarization in radiation therapy. I want to thank Ernie Brueggemann, who did all of the mass spec that I showed you, because I think that was very important in parsing out mechanism. Carol Colton's group at Duke, she had the Alzheimer's model that we used. And then everybody in Dave's lab, we had a lot of high school kids come through the lab, and they actually did a lot of the work that I showed you today. So I'm very thankful to them, and I wish them well in their futures. So thank you, and I'll try to answer any of your questions.
So what about EMT transitions?
Well, again, Jenny showed that a lot of EMT markers like twist and slug, those markers are up in her mice, in her mouse model. And I think she showed that they were also, I don't know if she showed that they were up in the patients, but I do know that she showed that they were up in her mouse model, and she could suppress them with NOS inhibitors. And interestingly, TIMP-1 is also an EMT marker. So if TIMP is high and can be modified in the presence of high NO, high NOS2, NOS2-derived NO, then that may also be promoting EMT as well. So
Consistent with your theory.
Yeah, it is. That's always good.
Any other questions? Okay, well, I don't see our speaker for 5 o'clock yet, so we'll take a 10-minute break at this point. Let's thank Lisa again. Dr. Pacher has arrived. He got his medical degree in Europe and subsequently came to the US. He's one of the world's experts in cannabinoid physiology, and he's in the Laboratory of Cardiovascular Physiology and Tissue Injury. He's a section chief and a tenured PI at NIH. He's at the NIAAA at Twin Brook off campus. The title, Interplay of Oxidative Stress, Inflammation, Lipid and Cell Death Signaling Pathways during Tissue Injury and Regeneration in Vivo, Implications for Physiology and Pathology. Al.
Thank you very much, Terry, and I thank you very much for coming to this seminar or listening. Just a second. Okay, we'll probably. Okay, and this is the pointer. Okay, sorry. So today I would like to talk to you about the complexity of the oxidative stress in vivo compared to various in vitro or ex vivo system, and how important to understand how oxidative stress is linked with inflammation, various cell death signaling pathways, and also various lipid signaling pathways. So first, this is a very schematic scheme which will be in the center of my presentation, and with different targets I will arrive to it back. So during any kind of tissue injury, you have increase of reactive oxygen species generation. Different sources may contribute to this increase, like mitochondria, NADPH oxidases, xanthine oxidoreductases, also uncoupled inducible or endothelial nitric oxide synthesis, and cyclooxygenases, among others. So when, for example, superoxide is generated through these sources, it also interacts with nitric oxide, which leads to generation of various reactive nitrogen species, including peroxynitrate, which then decompose to lots of reactive nitrogen species as well. And finally, this reactive oxygen and nitrogen species generation leads also to mitochondrial dysfunction and also oxidative DNA injury, which can trigger various cell death pathways, like polyadipylibose polymerase, which is a necrotic pathway primary. So usually this is the most abundant nuclear enzyme. It is triggered by oxidative stress or radiation-induced DNA breaks. It leads to NAD consumption and also ATP consumption, and it leads to cell death. But it also ribosylates various transcriptional factors which change the charge. They will be more negative. They can move, interact with each other, and therefore it is also a potent activator of various transcription factors, like nuclear factor kappa B involved in the inflammation, and it can also interact with mitochondria. This is Warren-Duffson science paper, where the small polyadipylibose polymerases can go to the mitochondria and induce apoptosis inducing factor release. And finally, the necrotic cell death and induce also secondary inflammatory reaction and attraction of various inflammatory cells, which then can also generate various reactive oxygen and nitrogen species contributing to this vicious circle. So today I would like to show you as an example, as a model of ischemic reperfusion injury, and go to some of these interactions of reactive oxygen and nitrogen species, their sources, and as well as with lipid endocannabinoid signaling, and show you what is happening and why the timing is also extremely important, which is very often ignoring targeting the injury. So usually ischemic reperfusion injury is the main mechanism of myocardial infarction, stroke, organ transplantation, vascular surgeries. When following the occlusion of the vessel, the reocclusion occurs. which leads to surge of reactive oxygen and nitrogen species generation and injury. But for example, if you have a myocardial infarction, it is inevitable to induce reperfusion injury because you need to remove the blood clots, and actually the sooner you remove, the faster the tissue will recover. So this is also inevitable consequence of the treatment. So again, when this happens, it leads to activation of various pro-inflammatory factors in endothelial cells first, which is the first defense layer. Then they release various chemokines that attract inflammatory cells, which produce then various reactive oxygen and nitrogen species, inducing cell death, and that leads to further injury. So it is very important when cells die, so when they die from necrosis, where there is massive tissue injury, they release various damage-associated molecular patterns, which are, I will have a next slide, which can be DNA fragment, it can be various protein like HMG Brooks protein, which is the most known, or even can be various oxidative stress byproduct, like byproduct of lipid peroxidation, like H&E, which is also used as a marker, so it can be labeled in the cells and measured. So if this happens, it triggers a secondary pro-inflammatory reaction, because this damage-associated molecular pattern bind to certain receptors on inflammatory cells and trigger inflammatory response. If it goes to apoptotic way, this is not triggering release of this damage-associated molecular pattern. So usually these cells are eliminated by phagocytosis or various other processes which do not lead to secondary inflammation. So this is an example of various damage-associated molecular pattern which act on different receptors. Like one of the most known is HMG box protein 1. This activates the same TLR receptors, like for example the endotoxin, but ATP can also activate receptors, various cytokines like IL-1 alpha, and DNA, RNA fragment, and even uric acid crystals. So, for example, during the gout, this is when uric acid is accumulated in small joints. Suddenly, after massive consumption of meat, for example, it cannot degrade properly, and these uric acid crystals can accumulate in joints, and it causes very bad pain and inflammation. And for example, this can also trigger NLAP3 activation in inflammatory cells. So going back to the model, I would like to talk about which today, and just outline in a relatively simple way how these processes interact, is a liver ischemia reperfusion injury model. So what happens that for 1 hour the main vessels for the liver are occluded, so basically there is ischemia. So during this process there is mitochondrial dysfunction and also endothelial dysfunction, and later these cells, hepatocytes or endothelial cells, start to die. So this is predominantly necrotic cell death at the beginning, and they start to release various damage-associated molecular patterns, and they activate the Kupffer cells. These are modified macrophages which are sitting on the sinus endothelial cells in the liver normally. And once these cells are activated, they can overexpress various NADPH oxidases, which then would produce superoxide or hydrogen peroxide, as well as inducible nitric oxide, and this diffusion limited reaction of NO and superoxide, you will have peroxynitrite. And also these damage-associated molecular patterns attract neutrophil to the site of injury. But as you see, during this first 2 hours, early, there are no much neutrophils yet, but there is the first inflammatory reaction, which is the activation of Kupffer cells. And the neutrophil attraction and infiltration occurs after 6 hours and peaks at 24 hours, where you see a very nice demarcation of necrosis. I will show you slides very soon. And this is also important because you will see that the inflammation goes up acutely, but it goes down, like the hepatocyte necrosis markers go up and they go down. But at later phase, you have massive secondary wave of the reactive oxygen nitrogen generation, and you have lots of apoptotic cell death, which normally at the beginning you have necrotic cell death, and later you have also different cells like macrophages coming, which are involved in tissue repair. So here is the time course. So this is a normal liver. So this is exposed to 1 hour ischemia and 2 hour and 6 and 24 hour reperfusion. So you see various markers of cell death. So first, these are liver transaminases. So these are indicators of liver function. And when hepatocytes die from necrosis, they are released from the cells. So these are also measured clinically in liver diseases. So you see that both of these transaminases peak around 6 hours. So the necrosis peaks at 6 hours, and at 24 hours, this is already going down because that time necrosis is not the predominant cell death. That time it shifts to apoptosis. And so this is this nuclear enzyme polyadipyribose polymerase, which I showed you at the first slide, which is a downstream effector of oxidative DNA injury. So it is increasing with the time. And DNA fragmentation, this is a marker of apoptosis, and caspase 3,7 is apoptosis. So you see that at 2 hours, you don't have apoptosis at all. And this starts to be more significant at 24 hours. So even the cell death is shifting. So here you see electron microscopy slides. This is a normal liver. So you see nice normal electron-dense black mitochondria. And 2 hours after reperfusion, you see swollen mitochondria with decreased structure. And you also see at 2 hours decrease of mitochondrial complex 1 activity. And you also see increase of lipid peroxidation. So this, as I mentioned, this H&E is a marker of lipid peroxidation. This is like a monondialdehyde, but this is a more stable, 4-HN is a more stable, more reliable marker, which can be detected immunohistochemically. There are ELISAs, which is pretty reliable. And also, there is also increase in carbonyl radicals at 2 hours from the isolated mitochondria, and also nitrotyrosine, which is a marker of protein nitration and also in part peroxynitride generation. So these markers early go up and you see mitochondrial dysfunction. But later, actually, by 24 hours, mitochondrial dysfunction completely recovers. And if you see the markers of lipid peroxidation, they start to go up further very significantly. And also, this is GP91 FOX. This is an isoform of NADPH oxidases. This is an OX2, which generate superoxide. So you see that at 2 hours, there is only relatively little Roche generation lipid peroxidation. So these are the mitochondrial part. And then later it goes up. And that's why it is extremely important to separate some in vivo experiment from in vivo. Because, for example, if you take a tissue, and most of the previous studies on which drug developments were based, and those practically completely failed in ischemia reperfusion, were isolated tissue preparation, like an isolated heart. They subjected to hypoxia, which was they just removed, they just put nitrogen in the Krebs solution and perfused. And after, they just reintroduced high oxygen containing, like carbogenated solution, which contains 95% oxygen and 5% CO2. And then they induce sort of ischemia and then reperfusion. But in those models, actually, the surge of this reactive oxygen nitrogen species happened right after the introduction of this oxygenated solution, and it lasted for very brief periods, for seconds or 10 seconds, extremely brief period. But as you see here, in vivo, the situation is totally different, and in those models, you also don't have inflammatory cells, and those are extremely important in either mediating injury or in the repair. So here you see normal livers stand for malondialdehyde, so you see marked increase in staining in these livers exposed to 24 hours. So this is this time point. So this is high magnification. You see it in both hepatocytes, and also here you see attached inflammatory cells, and they are attached to the activated endothelium. And this is a marker of oxidative DNA injury, 8-hydroxy-DG, which is, you see here, this is higher magnification, more clear cut. So you see increased staining in the nuclei, because this is, as I mentioned, a DNA injury marker. So the staining is localized in the nuclei of injured hepatocytes. Again, if you go back to the time course at 2 hours, nitrotyrosine going up, but relatively little, and this is coming mainly destructive oxygen nitrogen species generation from the mitochondria, but later this increase is considerably larger, and this is more magnification, so you see it everywhere. At larger magnification, you see that it is in hepatocytes, it is in endothelium, and here you see nicely this brown stained neutrophils. They are actually just coming, attaching to the endothelium, and when it is injured, they are just migrating to the tissues. And here you already see that from this damaged endothelium, which is endothelium black here, but here all the damage, they just go in the tissue to hepatocytes and mediate injury. So concerning the inflammation, and again, it is very important to distinguish cellular inflammation and inflammatory reaction, which, I mean, you can measure by measurements of various cytokines or chemokines. So for example, when you have neuroinflammation, you can have just microglia activation and increase in markers like TNF-alpha or other inflammatory markers without a single infiltrating immune cells, because this infiltrating cell needs to pass the blood-brain barrier. So in this case, you see that there is an early increase of various chemokines like MIP1-alpha and MIP2 at 2 hours. This is coming mostly from activated Kupffer cells, which are modified resident macrophages in the liver, and they play an important role. But later, it is going back. And the same with TNF-alpha, and this is adhesion molecules expressed in tissues, so different other adhesion molecules on endothelium. So when they expressed these inflammatory cells, so here you see nicely, so there is a vessel, lots of inflammatory cells, and they are just crossing endothelium and going to the tissue, so here from this vessel. And again, at 2 hours, you don't see any inflammatory cells in the reperfused livers. At 6 hours, they are already going in, and at 24 hours, you have a huge number. But despite the large number of neutrophils, you see decrease on inflammatory markers, because these neutrophils are primarily generating reactive oxygen species, not not pro-inflammatory cytokines and chemokines. So now a little bit summarizing, so what happens during this process, a little bit more detailed. So you have ischemic injury, which is when, for example, in specifically in case of liver, when you have a liver surgery or you have a liver transplantation, and when you they put back this liver to a person, they open the vessel and oxygenated blood is coming. And during this ischemia, you have tissue injury, release of this damage-associated molecular patterns which activate Kupffer cells. Activated Kupffer cells express NADPH oxidases, iNOS, COX-2, and generate reactive oxygen nitrogen species. They also secrete chemokines to attract neutrophils. This damage-associated molecular patterns directly also can attract neutrophils. And then finally, endothelium is also activated. They express adhesion molecules, and these immune cells stick attached to the damaged endothelium, and then they transmigrate to the tissues when they're getting activated and mediate injury. And again, I don't want to repeat the time course, but again, you have first initially an inflammatory reaction, and later you have a secondary and the mitochondrial ROS generation, but later mitochondria can even recover. And then this ROS is largely sustained by infiltrating neutrophils. So now I just show brief examples what happens if you start to target in this model various sources of reactive oxygen species, so for example, NADPH oxidases, xanthine oxidase, or nitric oxide synthesis, or mitochondrial ROS. So I'm sorry, I changed a couple of slides and I updated, but I can send you all this presentation, or Terry can send you. So most of the slides in the presentation, but there are a couple of new ones, and there is a slide reorganization. So anyway, first let's go what happens if you now there are different antioxidants which are possible to target to the mitochondria, and actually now this is a very fashionable approach to you can basically almost send everything in different organelles. For example, different antioxidants or NO donors or other drugs. And so, for example, we and others used a couple of these compounds and try to determine what is the clinical relevance targeting this process. And basically, if you give mitochondrial targeted antioxidant before the reperfusion injury, before ischemia, they are very protective, and they can stop almost all this process because they can stop the initial hepatocyte cell death. However, if you start to give them after the reperfusion, they extremely quickly lose their efficacy. So clinically, they are not very useful, because usually when you have reperfusion injury, like after myocardial infarction or stroke, you already have reperfusion for a certain period of time, so you cannot pre-treat these patients. And this is very important because, as I mentioned, mitochondria are very important in triggering this injury, mitochondrial ROS, but later all this inflammation and tissue injury is maintained by inflammatory cells. So that is also another significant distinction between ex vivo and in vivo models, and why lots of compounds based on ex vivo models like Langendorf preparations in the heart completely failed in clinical trials because they had no effect on the secondary phase. So a couple of other examples, what happens if you target inducible nitric oxide synthase or endothelial. So if you target endothelial, so if you inhibit endothelial, you actually induce vasoconstriction, and you induce more hypoxia, and you induce more injury. But if you target inducible nitric oxide synthase, which plays a role in the later phase of reactive nitrogen generation, you may have protection. But again, the problem with this that this similar to mitochondrial antioxidant, they have very limited use because they are mostly effective at early stages. So NADPH oxidases, there are several studies showing that they play important role in hepatic ischemic reperfusion injury and other forms as well. And they also induce, because this reactive oxygen species can induce activation of nuclear kappa B, which also induces TNF-alpha production. But again, the problem with this that they could achieve the greatest benefit when they simultaneously targeted NADPH oxidases and as well as inflammatory processes. So targeting NADPH oxidases by themselves is good if you do it very early, but during the later reperfusion phase, it has relatively limited value. Similarly with xanthine oxidoreductases, there are lots of different controversial studies. Some people find benefit, others could not find. Similarly with reactive nitrogen species, peroxynitrite, various peroxynitrite scavengers are very protective. Actually, they are more protective than all the above approaches in various forms of ischemic reperfusion injury. But usually they have other problems, that they are metalloporphyrins and very difficult to synthesize these compounds. They can interfere with clinical assays because they are usually making like urine blood green or black. So even though they are very effective, and some of them were also in clinical development for neurodegenerative diseases, they are very difficult drugs, and for reperfusion injury, again, the sooner you give, the more effective they are. And I'm sure that you have heard about cyclooxygenases during the course of this, the presentations earlier. And cyclooxygenases are also play very important role in reactive oxygen nitrogen species generation. And the reason why I'm also mentioning this, because when I talk about some endocannabinoid lipid signaling, there will be also very strong interactions with cyclooxygenases. So usually in the liver, blockade of cyclooxygenases is protective, but here you have to again think about it, that you inhibit generation of lots of prostaglandins like thromboxane A2 is very bad in ischemic reperfusion, but prostaglandin E2, for example, plays important role in liver regeneration. So and you inhibit that too. So basically, for acute reperfusion injury, inhibition can be good, but for chronic disease, it can actually adversely affect the whole system. So now I will talk a little bit about lipid endocannabinoid signaling, and also how it interacts with oxidative stress and with the processes, and also this is a lipid signaling. But a little bit introduction. I'm sure that you already know, you heard a lot about marijuana, which has anti-inflammatory, antioxidant effect, but also psychoactive properties for which it is crucified. But just to know that there are approximately 500 chemical compounds in the marijuana, and for the psychoactive effect, mostly this compound, delta-9 tetrahydrocannabinol, THC, is responsible. So this has effect on two cannabinoid receptors. There are two receptors. One, CB1, which is mainly in the brain. So it induces the psychoactive effect. It also increases appetite. So when people smoke marijuana, they also want to eat. And this also led to the development of CB1 antagonists for obesity. which were marketed in actually in large number of countries, almost all over Europe and more than 40 countries. But they introduced anxiety, and they finally were withdrawn. But they were very effective in diabetes, diabetic complication, and they were also effective in obesity. But the problem that CB1 receptor is the most abundant G protein coupled receptor in the brain. So it is on every single synapses. So it is inhibitory, so it can inhibit both excitatory or inhibitory synapses, and depending on the ratio or disease or condition, it can have lots of effect. But as I mentioned, there are lots of other compounds, and for example, cannabidiol was thought for many years that it is completely inactive, it has no biological effect. And at the time when Julius Axelrod and David Wink published that cannabidiol and tetrahydrocannabinol are neuroprotective antioxidant, nobody really believed that in the cannabinoid field. And actually, U.S. government has a patent for this, for which it is often criticized, but I don't want to go into this. But the bottom line that recently the mixture of these two compounds is used in patients with multiple sclerosis, patients with pain. It is approved in many countries with certain conditions, and it has certain beneficial effect. But more interesting is this cannabidiol, because it has no effect on either cannabinoid receptors, neither the psychoactive CB1, neither on the other one, which is anti-inflammatory. And so there are now probably thousands of studies showing beneficial effects in various models of inflammation. And usually the good about cannabidiol that it targets not only oxidative stress, but it is also a very powerful anti-inflammatory drug which target migration of various immune cells. And it is also very protective in liver ischemia reperfusion injury. And recently it is approved by FDA in children with epilepsy, and also for several orphan drug indications. And there are lots of about 40 different clinical trials with cannabidiol or mixture of cannabidiol with low doses of THC. So again, but the most interesting part is not that you have these ligands, which is in cannabis, but you also have an endogenous system, which is basically a lipid system. In every tissue, you have endocannabinoids, you have a different degree of CB1 and CB2 receptors, and they play very important roles, and they also may very importantly interact with reactive oxygen species generation, and also with arachidonic acid cascade and cyclooxygenases. So the two main endocannabinoids are anandamide, which is synthesized from basically membrane lipids, and also the second is 2-arachidonyl glycerol. So both may activate CB1 and CB2 receptors. 2-AG is more a ligand for CB2 receptors, and also more importantly, 2-AG levels in tissues are approximately 2-3 magnitude are higher. So it can be a very significant source of arachidonic acid when degraded. So you see that anandamide is degraded by FAH to arachidonic acid, and 2-AGs by MAGL to arachidonic acid. So I will talk briefly about these two enzymes. I don't want to go in all this. So basically, now there are tools to raise or decrease this enzyme, this endocannabinoid levels. By inhibiting this enzyme, you will raise anandamide levels. By inhibiting this, you raise 2-AG level, and you can also inhibit the synthesis of this compound. But I will talk primarily about this one, because this is the endogenous CB2 ligand, it is 2-3 magnitude higher in all tissues. So it can be a very significant source of arachidonic acid, which is a precursor of all prostaglandins. So as I mentioned, just CB1 receptors are in the CNS. They are responsible for psychoactive effect. They are also in the cardiovascular system, and they have very profound cardiovascular effect. And now it is a headline in top cardiology journals, the cardiovascular effect of marijuana, and particularly synthetic cannabinoids. It is inducing in children myocardial infarction. Synthetic cannabinoids are mixtures of potent CB1 agonists. You know well probably the spice and K2, or there are different names. Only 5-6 of these ingredients are banned. Usually this is a mixture of 5-6 synthetic agonists. But there are about 30 on the black market, and those you can buy in internet, and you can induce lots of harm taking them, because it can induce very profound cardiovascular effect. It can induce massive kidney injury. So kids are ending up on dialysis after smoking 1-2 doses of the synthetic cannabinoids. And there are lots of studies also showing that in inflammatory cells, CB1 receptor in macrophages, for example, can activate NADPH oxidases, also TNF-alpha release, and this contributes also to diabetes and different other complications, but I don't want to go in this. So the other receptor is the CB2 receptor, which has no detrimental effect. It is in immune cells, and usually CB2 receptor activation leads to decrease of chemotaxis of these immune cells, adhesion, and transmigration, and decrease of the pro-inflammatory response. And synthetic CB2 agonists are beneficial in various forms of ischemic reperfusion injury, and they are recently also under the development as anti-inflammatory medications for kidney inflammation, maybe CNS inflammation, neurodegenerative diseases, ischemic reperfusion injury. So now going back to this model, I will talk a little bit what is the role of this and how this endocannabinoid signaling also interacts with like arachidonic acid signaling and COX-2. So these are normal livers. Hemotoxin, I was saying this staining for neutrophils. So normally you don't have infiltrating neutrophils, so there is just one. So during the ischemic reperfusion, there is a massive necrosis, and here you see lots of neutrophils, which is decreased by a CB2 agonist, and there is increased injury in knockout mice. So as I mentioned, the two main endocannabinoids are anandamide and 2-AG. So during the ischemia, there is no much increase, but during the reperfusion in the liver, there is marked increase. And this is generated in basically all cells, in hepatocytes, Kupffer cells, endothelial cells. And also this correlates with the liver injury. But from this, actually one conclusion can be that these are endogenously generated to protect the tissues through CB2 receptors from further injury, or they promote the injury. So to address this question, I mean, various mice, as I mentioned, I'm just going back. So one degrading enzyme for anandamide is FAH. So if you inhibit urease anandamide, and for 2-AG, the MAGL. So if you inhibit urease, this, and you will have less arachidonic acid. So in the liver, you have CB2 receptors basically in the Kupffer cells only, so which are modified macrophages. In hepatocytes, you don't have CB2 receptors. So here what you see, that inhibition of the monoacylglycerol lipase, which raises the endocannabinoid 2-AG level, leads to, so this is ischemic reperfusion, massive necrosis leads to significantly less necrosis. And this is the neutrophil infiltration. So these are normal tissues, so just normal tissue treated with the drug. So it's chemoreperfusion, lots of neutrophils, and these are largely decreased by this. GZL-184, which is an inhibitor of this MOG-lipase. You also see increase at 2 hours, the early time point of various inflammatory markers, as I mentioned, NOX2, and then decrease. And NOX2 and the H&E goes up with neutrophils later at 24 hours, and also there is decrease. So I don't want to go in this figure in detail, so this is not that important. The important conclusion of this is that there were different knockout mice used for CB1 or CB2 receptors, and the conclusion was that the protective effect of this inhibitor of this MOG-lipase was partially mediated through the protective cannabinoid 2 receptors on immune cells, but only partially. So then what we are also curious what happens with the prostaglandins. So you see that, for example, during this chemoreperfusion, there is massive increase of arachidonic acid, which is decreased by MOG-lipase inhibition, because in this liver, it is a very important hydrolytic pathway. So which means that you raise 2-AG, which is through CB2 may protect against inflammation, but you also decrease arachidonic acid, which is a precursor of COX-2-derived prostaglandins. And different prostaglandins, prostaglandin E2 were increased, D2, thromboxane B2, which is a stable analog of thromboxane A2, which is very bad in ischemic perfusion. So these are basically COX-derived prostaglandins, which went up during the reperfusion injury, and they also decreased. And again, in various other models of liver injury, this is induced by endotoxin and galactosamine. This is a massive inflammation-induced model. You also see decrease of the injury. This is the liver enzyme, and also decrease of these prostaglandins. This is another model, and this effect was independent from affecting the cyclooxygenase 2 level, which generate the prostaglandins. So basically, here, how it comes together, you have an acute liver injury, which induces cell death in hepatocytes, and you have also the inflammatory response, which follows the cell death. You also have endothelial activation. So when you inhibit this enzyme, you raise the endocannabinoid 2-AG, but on the other hand, you also block the hydrolysis to arachidonic acid. So you will have less eicosanoids, which decrease inflammation. Most of these are generated in hepatocytes. And on the other hand, this 2-AG, this endocannabinoid 2-AG, can decrease the activation of endothelial cells and migration, and also attachment to the endothelium and the inflammatory response. So basically, we have something which acts like a combination of a CB2 agonist and a COX-2 inhibitor, which is targeting an endogenous system. But as I mentioned this is an acute liver injury model. And for example, prostaglandin E2 is very protective, and it plays very important role in liver regeneration. So blocking, for example, COX-2 with anti-inflammatory drugs, which you can buy over the counter and use for arthritis or pain, is not a good idea, because this can impair liver regeneration, and also a prolonged COX-2 inhibition can impair the kidney function. So for example, it is well known that in people who are suffering from arthritis, if they take COX inhibitors for a long time, they can have kidney injury, or even a normal person, because normally in the kidney, the flow is maintained by nitric oxide, but if you have kidney disease, your nitric oxide system is impaired. and prostaglandins are maintaining the blood flow. So if you take like olive or other COX inhibitors, so you basically decrease these prostaglandins, and you will decrease your kidney flow, which can lead to kidney injury. And even in normal person, it is known that one of the side effects of this drug could be non-steroid anti-inflammatory drug-induced nephropathy. So again, going back to the hearts and endocannabinoids, so the reason why I am mentioning, because now inhibitors of the other enzyme, FAH, which I mentioned, this was a very appealing target for anxiety and for certain CNS diseases, because the idea was that if you give a synthetic CB1 agonist, it will induce psychoactive effect. You cannot give it. But by raising the endocannabinoid levels, which would hit the CB1, you will have much less psychoactive effect. This is a short effect, and maybe you can beneficially modify certain neurodegenerative diseases in which CB1 receptors play role, or for example, anxiety, pain, in which CB1 receptors are protective. But what happened with these clinical trials, that basically last year, this was by a French company, Bayer. So persons taking this drug, even in the phase 1 trial, some of them had brain lesions, inflammation, even one person died. And then when they went back to preclinical data, there were inflammation in the lung of dogs. There were lots of different problems with this drug. And so going back, so endocannabinoids, for example, in the heart, they may have effect through the CB1. So these are cardiodepressive effect, some pro-inflammatory effect. Through CB2, they may have protective anti-inflammatory effect, but normally CB2 is not expressed in the cardiomyocytes and endothelium. It is expressed in infiltrating immune cells. So if you have myocardial infarction, CB2 agonists are good to protect from the increased inflammation. But if you don't have inflammation, only oxidative stress mediated heart failure, they will not do anything beneficial. And CB1 is definitely bad. And most likely what happened with the Phi inhibitors also, that endocannabinoids had effect through CB1. And in addition to that, as I mentioned, they can degrade to various arachidonic acid metabolites, and some of the prostaglandins may have vasoconstrictory effects, so they can induce vasoconstriction in coronary arteries, and then you will have basically an acute vasospasm, which can lead to myocardial infarction. So it is very complicated, and this is also a lesson. So, for example, many years ago, we used this knockout mice in which the endocannabinoid anandamide level is significantly higher. So in different tissues up to 10-fold or several-fold, in the heart usually 3-fold. And initially, based on literature, we expected that it could be protective. But we used the heart failure model induced by an anti-cancer drug. And actually what we found that in this knockout mice, there was dramatically increased mortality. There was dramatically increased oxidative stress, red compared to the black, and also nitrotyrosine formation. This is H lipid peroxidation, cell death in the myocardium, and also surprisingly, in a model which is normally not associated with inflammation, there was massive increase of neutrophil infiltration. And I mean, again, this is a lesson that when you target something, you have to try to think about the whole physiology. So if you target something in the brain, you also have to think about heart and other organs. And for example, like it is also lessons learned from lots of clinical trials with COX-2 inhibitors. So for example, Celebrex, which was one of the most widely used COX-2 inhibitor for pain arthritis, and now it is still initially it was withdrawn because when the report started to come that this class of drugs, and later it was confirmed that all COX inhibitor 1 and 2 either, so all this ibuprofen, olive, I mean, everything what you can buy over the counter, it would increase cardiovascular risk. So in patients with heart failure, they are contraindicated, and now you take them by your own risk. Celebrex came back later with a label, but you have to be very careful if you have cardiovascular disease or kidney disease, or there is risk for myocardial infarction in your family. So basically, summarizing this endocannabinoid parts, that oxidative stress induces release or synthesis of these endocannabinoids, which is a lipid signaling system, which can profoundly affect inflammation through CB2 receptors, which is in immune cells and protective, and usually it exerts the opposite effect on CB1, which is pro-inflammatory. And it can also lead to arachidonic acid formation, because the levels can be markedly increased in tissue injury. So then you can have also show of different prostaglandins, which can induce, depending on the tissue and the time, important context-dependent effects. And finally, I would like to just show you also an example briefly that oxidative stress can also be beneficial, and actually in many contexts it is just an effect, a reaction to an injury, and chronic inhibition, like I also mentioned with COX-2, is not good. So if you have an acute injury, you can target certain pathway and decrease it, but chronic inhibition of this pathway, either lipid signaling or oxidative stress, is always dangerous. So this is, I mean, a relatively simple model. They just amputated the tail of this tadpole, Xenopus tadpole, and you see that they also express the rose-generating dye in these animals, and you see that with time, when the tail start to regrow, there is marked increase of rose generation, and basically, if they inhibited this rose generation, the tail growth was inhibited. So this is just an example. They use different targets like NOx inhibitor, non-specific inhibitors, and you see here that with certain non-specific inhibitors of NADPH oxidases, the tail was not growing back. Like with vehicle, it is growing back nicely, and with these two, this is apocynin, non-specific NOx inhibitor, and DPI, this is a more general non-specific ROS inhibitor. So this is the same actually in humans when there were large-scale trials in diabetes, and they were trying to normalize everything like blood pressure and high blood glucose content. These patients started to die. So basically the conclusion was that drastic normalization of pathological changes is never good. So you have to, because obviously there are lots of compensatory mechanisms which are working, and you may interfere with these two, and they induce more damage than benefit. So, and finally, this is just my conclusion, that it is very important always to think about the complexity. So you may have a brilliant new target targeting whatever signaling process, but also try to think what happens, what could be the role of this signaling process in other organ systems. And this is very important because, like most of the drugs, actually, magic bullet in a few years usually fail, and they are retracted because it turns out that they have side effects in other organ systems, like most commonly cardiovascular, like it happened also with some anti-diabetic drugs affecting PPAR signaling and so on. So, and finally, I would like to thank you for listening this and coming to this presentation, and our scientific director, my mentor, and different members of my lab who contributed to some of these studies. Thank you very much.
Any questions for Paul? So in terminal cancer patients, they often use opioids. Would it be better to use cannabinoids?
This is still controversial. I mean, depends for what because I mean there were lots of studies showing benefits of cannabinoid and marijuana in cancer, but there were also other studies showing exactly the opposite. And actually, the tricky thing that most of the studies were done in cell lines, and usually when they showed the positive effect, those were in micromolar range. But there were some studies showing dual effect, and they showed the opposite effect in nanomolar range. And again, I mean, the first studies showing in a few patients in glioblastoma multiforme, which is one of the worst brain tumors, which is very malignant. It is almost impossible to cure. It is always reoccurring, and the prognosis is bad. So they showed that they injected cannabis, THC, in these tumors, and they shrunk. But this was many, many years ago, and nothing still happened, and nobody ever could reproduce it. If it would be really that good effect, I think everybody would jump on it, and because this is, for example, a tumor for which there is practically nothing. But in case of cannabidiol, which I mentioned during my talk, which has no effect on psychoactive CB1 receptors, it has antioxidant effect. So in that case, actually, lots of studies, they are showing anti-cancer effect, and not a single study showing the opposite. So this is, I would say, significantly more promising, and this was also approved by FDA for glioblastoma multiforme, but like for a pilot sort of study for rare disease. This is not a big study yet. So I think it still remains to be seen. But I think for cancer pain, low dose of THC with cannabidiol or cannabidiol alone in certain patients who do not react to opioid well could be an alternative, because some patients, they react actually very well to cannabinoid, but others do not react at all. So usually this Sativex, called Sativex, this is an oral mucosal spray, alcoholic spray, which contains THC and CBD. THC very low amounts, and with this oral mucosal route, the levels will be very low, so it will not induce a very minimal psychoactive effect. So they usually titrate in these pain patients first if they are responding to this. So if they are not responding, they are not doing further. If they are responding well, they try to titrate an optimal dose, which is keeping the levels still low, but effective. But I mean, again, in some patients, even cancer patients who has terrible pain, nothing else is working, and they are reacting well. But I mean, I think still lots of studies need to be done, and the major problem with like marijuana that now in lots of states it is legal. So you have this marijuana dispensaries where you can buy for anything, but you have no idea what is in that marijuana. And this makes huge difference, because actually now if you check the trends, like past 10 years, the THC content, which is the psychoactive ingredient, is like going up. And also the other thing that, and again, and it can induce cardiovascular effect. And now the cardiovascular incidence of cardiovascular effect also going like this. And now this is a big problem also how to control. And the second problem, that it is impossible to do clinical trials, because everybody, you don't know what is in that marijuana. You can buy in the dispensary 10 different things. you have no idea what is there. As I mentioned, there are 500 constituents. You will never know if the beneficial effect was because of CBD was high. Maybe THC is not even there, or just it was THC and the person felt because of the high that he improved, but there was no real improvement. So, I mean, and the other problem is that also how you smoke it, it can dramatically affect what you get in, because if you smoke at certain temperature, you will get the good part like CBD and THC, but if you smoke at different temperature, you will not get any of this, but you can get all the bad things like from tobacco, all these carcinogens, everything. And in fact, marijuana is very well known that it can accumulate lots of cadmium and lots of heavy metals. So if it is growing somewhere there, you can get lots of environmental toxins as well. So, I mean, again, this is very complicated, and I think it definitely has to be regulated, because otherwise it will be just a huge mess, and eventually everything will just explode and will lead to nothing.
Still an area that needs a lot of work.
Yeah, exactly.
Okay, I think in view of the late hour, we'll adjourn. Thanks again for coming, Val.
Yeah, thank you very much.